welcome to this next video in OCR A-Level Biology. This is Module 2.3, which is Nucleic Acids. Now, this is the first video in a three-part series on this module, and in this video, we're going to look at the structure of DNA and RNA. So we all know that there are four types of biomolecules: carbohydrates, lipids, proteins, and nucleic acids. Carbohydrates, lipids, and proteins we covered in the last module, which is biomolecules. In this module, we're going to be looking specifically at DNA, RNA, how DNA replicates, and protein synthesis. So, I mentioned in the last module that all biomolecules have some sort of monomer. And the monomer for nucleic acids is a nucleotide, and that's what's shown here. So this is a diagram showing a nucleotide. So let's label this nucleotide. So here we have a simple diagram of a nucleotide. Excuse me. So here we have a simple diagram of a nucleotide. The orange thing here, this is our sugar. And the sugar is normally deoxyribose or ribose. It's a pentose sugar. To the right, bonded here to the sugar on the right, we have our nitrogenous base, or just base for short. And the base can be made up of one of five, and I say five, one of five different bases. Adenine, which is represented by an A. Guanine, which is represented by a G. Cytosine, which is represented by a C. Thymine, which is represented by a T. And uracil, which is represented by a U. Now, adenine, guanine, cytosine, thymine, you'll find these, your adenine, guanine, and cytosine, you'll find them in both DNA and RNA. Thymine, you'll find only in DNA, whereas uracil, you'll find only in RNA. To the left of the sugar here, we have our phosphate group. So here we have our PO4 minus. And it's these phosphates, because they're negatively charged, they are what give DNA, RNA, or nucleic acids a negative charge. So nucleotides, they are the monomers of nucleic acids. And when a nucleotide is made, there are condensation reactions which take place here, where condensation reactions take place, removing water in the process. So these are condensation reactions. Actually, I should have made that a bit of a different color. Whoops, I didn't mean to remove that. H2O, which is where this OH, this H here, and this OH come together to make this H2O. And then here, this OH and this H come together to make this H2O in our condensation reactions. So condensation is producing water. And here we can see that we've produced our nucleotide. So we have our 3 different building blocks of the monomer, the phosphate combining with the sugar to form the black water here. It's not really black, but in here is to denote that this is when these two come together, it makes this water molecule. And when the purple rings come together, these ones and this one, they form this water when the base joins with the sugar. And that forms this, which is our nucleotide. Now, so that's just this basic thing, all out of the way. Now I'm going to talk about something called complementary base pairing. So in DNA, bases pair up using hydrogen bonds. So bases pair up using hydrogen bonds in DNA. And this is called complementary base pairing. Complementary base pairing. And complementary base pairing is when when one nucleotide pairs up with a specific nucleotide on the other with a okay. Complementary base pairing is when one specific nucleotide pairs up with another specific nucleotide and it stays like that. What I mean is, let me give you the example of adenine. So adenine is a base, okay? Its complementary base pair is thymine. So A will always go with T. Likewise, C will always go with G. And this is in DNA. Remember, we're talking about DNA here, not RNA. A will pair with T via the means of 2 hydrogen bonds. 1, 2. C and G will always pair up by means of 3 hydrogen bonds. So these blue lines I'm drawing here, these are hydrogen bonds. Now, when we talk about complementary base pairing, we often say purine to pyrimidine. So what are purines and what are pyrimidines? Well, the A and G, adenine and guanine, I'm going to label them in red. Adenine and guanine, these are the, let me double check. Let me double check. Adenine and guanine are the purines. So adenine and guanine are the purines. A, G. And what this means is that they are made up of 2 rings. They have 2 rings in their structure. That's all you need to know. Thymine and cytosine, which I will label in purple, so these are known as the pyrimidines. Pyrimidines. And uracil as well. So thymine, cytosine, and uracil as well. And these have 3 rings. The way I like to remember that is that the purines are A and G, pyrimine CUT. What do you get? What made up word do you get? You get AG cut. Well, that's how it's a very weird way of remembering it, but it's how I always memorize it. That A and G, these are the purines. CUT are the pyrimidines. I also remember here, the reason I remember like this is because also you can remember it like 2 rings, 3 rings, purines. This is 2 rings here. And then pyrimidines 3, 2, what comes first? 2 and then 3. So AG cut. There you go. It's a really weird way of memorizing it, but that's how I memorize it. Okay, so this is, but remember, this T we're talking about in DNA. What about in RNA? In RNA, we swap out the T for a U. So this is only in RNA. Whoops. So that's complementary base pairing. Now we're going to talk about the DNA structure itself. So DNA is a double-stranded molecule. It's made up of two strands, each with a sugar phosphate backbone. So the sugar phosphate backbone is basically this structure here. The phosphate joined to the sugar, joined to the phosphate, joined to the sugar, so on and so on. And these are linked together by something called phosphodiester bonds. Where is it? Which one? Yeah, phosphodiester bonds. They're linked together by phosphodiester bonds. I'm just double checking. Yeah, phosphodiester bonds. So these are phosphodiester bonds. So they're held by phosphodiester bonds. And when you have this, lots of phosphodiester bonds, you call it the sugar phosphate backbone. So this yellow structure you can see here is called the sugar phosphate backbone. Here, this green bit is a complementary base pair. So these are basically complementary. So here we have complementary base pairing happening. Complementary base pairing. Pair. And oh, this diagram is slightly wrong. So here we have phosphodiester bonds forming the sugar phosphate backbone with complementary base pairs in the middle. Now, I mentioned above, this diagram's wrong, that A pairs with T using two hydrogen bonds, like this. C pairs with G using three hydrogen bonds. And this is the base pair. And this, so here we have the sugar, and in the sugar in DNA, remember DNA stands for deoxyribonucleic acid. Deoxyribo means that the sugar here is deoxyribose. So the sugar here is deoxyribose. And the phosphate here, this is the phosphate group. And here we have one base. Together, these three come up to form one nucleotide. So what I'm drawing the circle around now, because it's not very clear here, this is one nucleotide. Nucleotide. Now, we say that the DNA strands are anti-parallel. What does that mean? Anti-parallel means that they run in opposite directions. And you can clearly see that one is going up this way and the other is coming down this way. So they run in opposite directions. The one here, the one on this side, runs 5 prime. The 5 prime end is at the top here. What do we mean by 5 prime? Well, here we label the carbons after the oxidant. If I go back up here to show you, and if I go back up here to show you what I mean by 5 prime, here the sugar, let's call it deoxyribose, is labeled on the sugar, the carbons are labeled right after the oxygen, in a clockwise manner. So here, this is the oxygen, and we label the carbons as 1 prime, 2 prime, 3 prime, 4 prime, and the 5 prime carbon is out of this ring. So you number it right after the oxygen in a clockwise manner. That's the same thing we're going to do for the whole, so that's for one nucleotide. Now we're going to do the same thing for one nucleotide here. So as I said, here we start at the oxygen, which I'm going to mark with this dot. And then here we have 1 prime, 2 prime, 3 prime, 4 prime, 5 prime out of the ring. So we say for this strand runs 5 prime to 3 prime. As you can see, 5 prime to 3 prime. So we say 5 prime to 3 prime direction for this strand. This strand, let's do it again for this same strand, but keep in mind that DNA strands running are anti-parallel. Okay, so let's do it again after the oxygen in a clockwise manner. 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, and the 5 prime carbon is outside of this ring. Okay, so this strand, seeing as it's here, so the 3 prime end is at this top, and the 5 prime end is at the bottom. You can see here very clearly here. 3 prime is at the top, 5 prime at the bottom. So what we say for this strand is that this strand runs in the 3 prime to 5 prime direction. It is a little confusing as to, we say they're running in opposite directions, but here we're drawing 5 prime to 3 prime direction this way, here 3 prime to 5 prime direction this way. But if you think about it, if you have this is the 3 prime to 5 prime, if we say everything is 5 prime to 3 prime, for instance, then this one will be going down like this, so this will be going down. But if we put the 5 prime to 3 prime direction on this strand, what we can see here is, let me just denote it with a dashed line, that it's actually going in the other direction. If I zoom out, we can see it's actually going in the other direction. So this way is 5 prime to 3 prime for this strand, but this way is 5 prime to 3 prime for this strand. So we say to denote them going in the same, what's the word, same direction. If we want to make it in the same direction, when they're not, they're actually going in opposite directions. Here we say 5 prime to 3 prime in this direction, in the downwards direction. But here, for in the downwards direction, we say 3 prime to 5 prime. Hence they are opposite directions. Hence in the upwards direction, this one is 5 prime to 3 prime. But in this one, this is the 5 prime to 3 prime in the downwards direction. Hence we can conclude that DNA strands are antiparallel, they run in opposite directions. It sounds confusing, and it was quite confusing for me when I first studied this. But that's just the way you gotta learn. So that was DNA, the structure of DNA. I'm gonna very briefly talk about RNA. So RNA is similar to DNA, it's also a nucleic acid. But it has a different base to it. It has some differences. So here I'm going to draw a single strand of RNA. So this is one strand of RNA is generally, so here's a few exceptions. So this is RNA. So here we have A, I'm going to use different colors, G, C, and then the different one is U. So that's one major difference between RNA and DNA. one of the bases is different. So U replaces T. So U itself replaces thymine. That's the major difference. Another major difference is that RNA is the sugar in RNA. In RNA stands for ribonucleic acid, which means that the sugar in the sugar phosphate backbone, that sugar is, it's also a pentose sugar, but its name is ribose. So that's another major difference. Another difference, a third difference, it's not so major because there are some exceptions, is that RNA is generally single-stranded. Yes, whilst DNA is generally double-stranded, there are a few exceptions where you can have double-stranded RNA or single-stranded DNA, but generally RNA in eukaryotic cells, RNA is single-stranded. And there are multiple types of RNA. So there are multiple types of RNA, which we will discuss in our protein synthesis video. So there's mRNA. So these types of RNA we'll discuss in our protein synthesis video, rRNA and tRNA. So these are all used in protein synthesis, which we will mention in our protein synthesis video. So they're all useful in protein synthesis. But we'll mention this in our protein synthesis video. One last thing before I finish up is that how DNA can be packed into chromosomes, into one nucleus, how DNA is packaged in the nucleus. Just very briefly, here we have our nucleotide. So here we have the building blocks. This is just a quick chart, a quick flow diagram displaying how we organize DNA into the nucleus. So here we have the three main parts to one nucleotide. We have our base, ATCG, our deoxyribose, and our phosphate. All three of these make up one nucleotide. Same thing here. All three of these make up one nucleotide. All three of these make up one nucleotide. Multiple nucleotides. There's more. I'm showing three here, but there's multiple. There's actually more than three. Multiple nucleotides make up the DNA molecule, which is our antiparallel structure, our double helix structure, twisted ladder, antiparallel. All of our DNA, one DNA molecule, is actually called a chromosome. One DNA molecule plus some extra proteins and stuff in which the DNA molecule is wrapped around. But we'll get to that later. Is called, so one DNA molecule, which comprises of the DNA itself with some proteins called histones. More on that in our module 6.1 video. It's all packaged up into one chromosome, as you can see here. So this is one chromosome. I'll draw a chromosome actually, just to illustrate this. So all of our DNA is packaged into one chromosome, and that's what this is. So this is a chromosome here. We have 23 pairs. Humans have 23 pairs of chromosomes, so 46 chromosomes in total, and those 23 pairs are all packaged into the nucleus. So you can see how our DNA, how all of these phosphate deoxyribose base, all of these components form nucleotides. All of the nucleotides come together to form DNA. DNA plus some histone proteins come together to form chromosomes. All the chromosomes come together to be packaged in the nucleus. So you can see how we can get from DNA up all our DNA into nucleus. And we have over 2 meters of DNA packaged into a tiny, tiny nucleus, which is impressive. For a very large amount of DNA into a tiny thing. Anyway, that's going to be it for this episode here. In the next video of Module 2.3, we're going to be looking at how DNA replicates. Thank you all very much for watching, and I'll see you all later. Bye.